Everyone. So a little different talk here than we've heard some great research and from the previous two presenters, and really my opportunity here to present is hopefully to further enable those type of researchers and to continue to build out the great science that our researchers do around plants. And let me tell you a little bit about Ferris Genomics. So Ferris Genomics has been around, we're all of 2 months old at this point, two and a few weeks. And we're really set up to get more data, get more samples through analysis, and we'll talk a little bit about that today. And when we span across a bunch of different platforms, or a bunch of different crops and species. No. Okay, yep, I got it. So first of all, a little about myself. So, is that yours? Yeah. Okay, a little about myself. So I've had a career, whether it was at pharmacy at Pfizer or later at Monsanto, of really being responsible for generating as much data as possible in order to make decisions, and whether it was pathways or if it was starting to think about breeding, PCR, TACMAN, et cetera, for marker-assisted breeding. And then later in career, really as the sequencing came on, to take full advantage of those tools in order to help support breeding programs at Monsanto. And so one of the things we went to go about with building out the team at Ferris is that our team is all a bunch of folks that have lived this world. They've had to provide data in order to help with a breeding program, or in order to meet peak, in order to understand the genotype associated with the crops in the field and put that with phenotype and think about the next planting season. So we've built out a group that has lived this, that understands that the responsibility of delivering data very quickly and with lots of data in order to support some of those breeding programs. We have offices in St. Louis, Missouri, and also in Limerick. So one of the key drivers for reason for Ferris Genomics is, just as the other speakers have said, the cost of sequencing has gone down tremendously over the years. In fact, we're nearing that $100 genome, and that's been a key driver to this. One of the main responsibilities at Monsanto was to, as much as I could, get as much data for as cheap as possible. It's not that we weren't going to spend the money, it's that we wanted more data. And the same thing is true. So one of the things that since the team that I had at Monsanto put in a genotyping by sequencing process, it's been about 15 years. And not much has changed. We've driven on a 1,500 to 2,000 marker for quite a set for quite a while. Now with the ability of the sequence getting cheaper, there's opportunity now to do more of a whole genome sequencing. One of the things that I've been intrigued about over the last couple of years has been skim sequencing and the ability to impute genomes, and then to utilize all that extra information to hopefully make better breeding decisions to be able to feed into our breeding pipelines. So as a lot of you may know, one of the major costs still associated with whole genome sequencing is the library prep piece. Over the years, part of what I've been able to take advantage, whether it was at my time at pharmacy at Pfizer or later at Monsanto, and then even later in career, has been to high throughput, high content screening. So our ability to take something at a big volume and reduce it to micro volumes. And so that's been a big part of our learnings over the years, is how to take something in macro scale and drive it to micro scale, but still get the same content. One of the things that we've been working on at Ferris, and part of this has been somewhat stealth, is we've been working on AMRA technology. And AMRA technology is the ability to enable a microfluidic miniaturization process. So we're able to increase, one of the interesting things about AMRA technology, although we're doing total volume reaction of about 1 microliter, we're providing most of the reagents at 100 to 200 nanoliters. That there's a little bit of special sauce related to our microreactor. The reaction efficiency is very high. We enable faster delivery of some of these quality reagents. It helps us deliver data, but probably most importantly, as you can imagine, as you reduce size, you can also reduce costs. You can get more sequencing done for your dollar, so you can get the library prep cheaper as well. So this has been a big mantra for the work that we've done. And so more the next generation, you know, the AMRA piece of this represents the potential to really do a shift. And I've been kind of very excited about this, the ability to generate more and more sequence data, especially at a whole genome level with imputation, and deliver that data to a breeding program in order to make better decisions in terms of breeding prediction. The way AMRA works is that we miniaturize the reactions. We've been able to, with our carrier fluid and with specialized coated capillary tubes, we're able to pull out the reagents and deliver them in droplets. We're able to do this in precise formula where we can pull up the DNA sample, and then all the reagents associated with full genome processing. We can then deliver this into our bioreactors and then run through the normal process of fragmenting, fixing A-tailing, and then later, obviously, adapters, et cetera, to get to a full library. So one of the things that's always been common in its early attempts of kind of droplet-based PCR, as well as ability to make library in droplets, has been that there's sometimes problems with this, of moving droplets under plastics or in tubes. There are certain phenomena that happen with leaving microsatellites and leading to contamination. One of the things that our team really understands is ways around this and how to do this properly. What I represent here is just the ability to carry droplets in tubes. What you're seeing there is basically a TACMAN reaction, where we've gone and dipped and pulled 10 non-NTCs, so no transcript there, no DNA, then ran a strip of 50, I just turned it off, there we go. A strip of 50 positives, followed by a break, then 50 more positives of a different, then NTC. And what you can see is that compared to cycle 7 with no amplification, by the time we get to cycle 42, we see the increase in signal compared that we should, and where MTCs stay low. So we know that we can carry lots of droplets through tubules without leaving microsatellites and leaving to contamination. One of the other interesting things about our technology is that when you start to deal with nanodroplets, it gets sometimes hard to pick them up out of plates and out of things. So you have to have special opportunities there. And so one of the things that we learned early on is that just dropping droplets into a PCR plate, they tend to stick to the side of the plastics. They tend to move. If anyone's ever tried to pick up a small volume of a PCR plate, allows static, pushes the droplet to the side, it's hard to get to. One of the neat things about our AMRA technology in terms of bioreactor is that with a little bit of change in the surface of the plastics, we can actually cause a droplet to levitate and center. And this is interesting in that it then allows us to easily access this. It doesn't move, allows us to drop in other reagents and coalesce with the droplet. It's also in a PCR plate, which is pretty common for thermal cycling, which means there really isn't a need for any other special plates other than what we provide here. So Complete Genomics, I think with the flexibility that the G99 and G400 provide us, we get very fast DNBSEQ technology for using the G99 for small genomes or for small sample sizes. It allows us to run these through relatively quick. Remember that a big part of what we're trying to do is run things through fast. We want to be able to return data in 5 to 10 days to really push breeding programs, particularly speed breeding, which is one of the things I'll be talking about. There's a lot of versatility. We have both the G400 and G99. Will soon be the T7. This allows us to run various different sizes. In order to deliver the low cost, you need to be able to put the right number of samples with the right depth of coverage on the right system. So we get a lot of flexibility. Also, the ability to run independent flow cells is pretty key to allowing us to run part of a flow cell on one set of samples and then bring another set of samples onto a different flow cell. Flexibility of the technology is incredible. It enables us to run those individual flow cells, as I was just saying. It's very accurate. So, you heard earlier from Wenqi that, we get greater than 99% SNP and indel precision sensitivity because of that rolling circle technology piece that, this is very, very good data. And then part of what we're trying to do, and luckily with some of the new sequencers coming on board, particularly Complete Genomics, it allows us to get to a cost-effective solution. So coupled with our AMRA technology, with the sequencing, we're able to drive down the cost considerably. So as I said, prior to both AMRA and DNBSEQ, technology costs were, about 60% of the total cost of running this was related to library prep. With AMRA technology, we're able to bring that down significantly. Couple that with sequencing technology, now we're getting into a price point which will enable a lot of us to run a lot more samples and hopefully get a lot more data. So one of the test pieces that I wanted to show was just speed breeding. So this has been a big part of what has driven a lot of the major ag to get successes in the field and to really drive yield and other traits. The history of speed breeding has really been, over the last, it's had a fine evolution in terms of getting there. Now we do a lot of our work in greenhouses. We're able to take our parents. We're able to do full sequencing of those parents, do our crosses, and then with the advent of kind of skim sequencing, we're able to then skim that cross, so it allows us to get a lot of samples through a sequencer, and then to get that data back imputated, and with now, instead of looking at a 1,500 to 2,000 marker panel, we have the ability to look at millions of samples and to really understand the imputation process. With that data, hopefully we're able to feed that into our predictive breeding pipelines and get a better choice of next crosses, et cetera, to advance. So at Ferris Genomics, our first part is sampling, and we provide a kit that allows you to very quickly do leaf punches. It makes it easier for us to flow the data through our process. Rapid and high quality DNA extraction is part of this. Like I said, almost all of our team has had a lot of experience doing this. So we understand what are inhibitors. We understand what kind of reagents we need to do to get a pure extraction in order to move this forward through the sequencing process, especially library prep. We utilize our AMRA technology to enable this high quality library construction at high efficiencies at much lower cost. We then can couple that with skim or low pass sequencing coupled with an imputation pipeline to get answers, and allows this really for us to do this in 5 to 10 days. This is important for some plants, for quite a bit of plants where you want to be able to turn these over pretty rapidly in the greenhouse that way you can make many decisions before you get to the field. My main job at Monsanto a lot of times was to make sure that the data and everything was turned so we can make decisions before we got in the field, because the field was where cost actually really incurs once we start to try to test things in the field. Data delivery, we can deliver FASTQ files, we can demultiplex those, we can return that as a BAM file or VCF file. There's many different options. We also have a partner that we announced today with GenCove. They have a nice imputation pipeline that can deliver data that you can then feed into your pipeline for breeding. One of the things that I've talked about a little bit, and we'll be generating more data. Like I said, we're only a few months old at this point, but we've understood this technology for a while. So I give you this example, and I would ask you all to kind of join us at 6:20, where we'll have some of our scientists present on a little more data around this in Pacific D. But we are able to take a 1x reaction size, so a normal controlled as the manufacturer suggested, and generate a soy library that produced a nice library that we were able to run. When we compare this with 1x with our AMRA bioreactor, get a very similar library produced. One of the nice things about the AMRA technology, as I said, was that it allows you to reduce this and also get very high efficiency. So what you're seeing in this next is that we went to a 10-fold reduction in size of the library. When we run it alone without the AMRA bioreactor, we get this type of coverage around a very uniform set. But with the AMRA technology, in terms of the bioreactor, we get a much better library produced. In summary, I just wanted to let you know that Ferris Genomics AMRA technology, coupled with Complete Genomics DNBSEQ technology, gives you a nice platform to really run more data with more flexibility, speed, and accuracy. You can get more data for your breeding dollars. And the power of this is that hopefully you can drive your predictive breeding programs. I want to thank Complete Genomics for giving me a chance to talk a little bit about the technology. And we hope to be able to share more as we continue to grow and move forward. I'll open up for any questions.